All righty. So this week, we're going to be covering chapter 15, which is antimicrobial drugs. So this is kind of taking everything that we've learned this semester, putting it all together, but looking at how we can target specific structures, okay? And how we can stop the functioning of the virulence factors that we don't want work doing their thing and that kind of thing. Okay. All right, so here's an overview of what we're going to cover today. We're going to go over the learning objectives. I'll open it up to questions, and we're going to review bacterial and eukaryotic cell structure with an eye toward reminding ourselves the differences between our cells and bacterial cells, because those are going to be targets for the different classes of antibacterial drugs. Okay? And then we'll talk about what to do for next time. All right, so this chapter helps us move toward being able to meet the following learning objectives: being able to define the key role of evolution as it applies to microbiology, identify microbial structures and connect the structures to their functions, identify necessary components and processes in microbial metabolic pathways, identify key agents and processes of microbial information transfer, identify pivotal components of microbial systems important to human health, and analyze and describe the impact of microorganisms. All right, so any questions? I have a very good question from Mitchell, so any other questions, I'm happy to answer them. Okay, if you think of any, let me know. All right, so we're going to review eukaryotic and prokaryotic cell structure. Specifically, we're going to go with bacteria. And we're going to go with a human cell, which is eukaryotic. Okay, so what I'm going to do is I'm going to have you all walk me through building a bacterial cell and a human cell. Okay. Now I realize it's been a long time since chapters 3 and 4, but as a reminder, when you struggle to remember, you remember the answer better when you get it. Okay, so if you don't remember, that's fine. I'm going to make you stew a bit. Okay, so give me a structure, either bacterial or eukaryotic.
A bacteria cell is going to have a cell wall.
I'm going to have a cell wall, yes.
Peptidoglycan, I think.
Very good. Okay, and I'm going to draw just part of a bacterial cell. And let's go ahead and put peptidoglycan. Okay, very good. Okay, is this a structure that human cells have?
No.
Nope, very good. So this is potentially a target for antibacterial drugs. Okay? What does the cell wall do for the bacterial cell?
Stops it from lysing, right?
Very good, yes, it stops it from lysing. Okay, water comes in because we've got a higher salt concentration in here, and pushes up against the cell wall, and the cell wall prevents it from lysing, prevents it from popping. Okay, very good. Okay, what's another structure?
The bacteria, actually, never mind.
They have a what?
I was going to say they have the vacuole, but
you know, vacuoles are mainly in plants.
Yeah.
Yeah, plants and protozoa. But what do all cells have to have? If they don't have it, they're not a cell.
Cytoplasm.
Yep, they all have cytoplasm. Okay, what holds the cytoplasm in?
Phospholipid bilayer.
There we go. And what do we call that phospholipid bilayer?
Cell
The
membrane.
lipid membrane.
Yeah, very good. Cell membrane. Okay, is that something that's only in prokaryotic cells?
Nope.
Nope.
So we do have it in our cells. Do we have a phospholipid membrane? Cell membrane?
Yes.
Okay, so let's go ahead and put that on both of them. And that's what keeps the cytoplasm in. Okay, it's a boundary between the outside and the inside. Okay. All right, what are some other structures?
The human eukaryotic cell is going to have a mitochondria.
Oh, okay. Yep. Let's go ahead and get a mitochondrium up here. I'm going to do just one. Okay. So it's got an outer membrane and it's got an inner membrane that's got these funky folds. Okay. What does the mitochondrium do for the eukaryotic cell?
Package energy.
Very good. And makes the bulk of the ATP. Okay. So what are the pathways, just for the fun of it? Let's go ahead and review the pathways that mitochondria have that eukaryotic cells wouldn't have. What are the pathways that make the bulk of those ATP?
Will those be the endoplasmic reticulum?
Nope, we don't have endoplasmic reticulum in here, but we do have endoplasmic reticulum in eukaryotic cells. And we have two kinds. Okay, what two kinds do we have?
Rough and smooth.
Rough and smooth. Okay, and what do they do? What does rough do? What is it studded with that makes it look rough under the microscope?
Got the ribosomes in it.
Very good, ribosomes. And what do ribosomes do? Let's go ahead and get some free floating ribosomes out here. Yeah, what do ribosomes do? They synthesize what?
Don't they
Protein.
bind RNA and transfer RNA?
Yep. And that enables them to do what?
Synthesize proteins.
Very good. Okay, so protein synthesis. Okay, do prokaryotic cells have ribosomes?
Yep.
They do. Okay, are there any differences between the prokaryotic ribosomes and the eukaryotic ribosomes?
70 and 80.
Very good. Okay. Now, having said that, do mitochondria have ribosomes?
Yeah.
They do. And what size are they?
70.
Very good. Yes, they are 70. Okay. Okay, do mitochondria have anything else that they share with their free living cousins out here?
Well, you've got your pili.
Mitochondria don't have pili, but let's go ahead and put some pili on this guy. pili do?
Attach.
Yep, they attach.
That's how they
Yeah.
transfer their information to their buddies.
It is. That is how they transfer their information to their buddies. Okay? But we don't need that over here. We're living very happily inside of a eukaryotic cell. We don't need anything to grab a hold of stuff. How did we figure out that mitochondria used to be free living bacteria?
Endosymbiotic theory.
It is the endosymbiotic theory, but we sequenced what?
DNA.
DNA, yes. They have a little circular chromosome here that's composed of DNA. Okay. Free living bacteria have bigger chromosomes because they've got to do more. They've got to make more proteins because they're living out in the wild. Hey, do eukaryotic cells have a chromosome? Or more than one?
Do they have what?
And do eukaryotic cells have chromosomes?
Yes.
Yeah, and where are they? In what structure?
Nuclear envelope.
Very good. Yep, in the nucleus. We have the eukaryotic chromosomes, and they're linear. And we'll do just 2 for the sake of time of drawing these puppies. Okay. All right, so let's get back to this mitochondrion. Pathways that mitochondria have. Aerobic bacteria have them too, but that make a whole bunch of ATP. One of them is
Aerobic.
cycle. They are aerobic pathways, but I'm looking for specific ones. One of them is the cycle.
Is it the citric acid cycle?
Very good. And what comes before that? What have we got to do to
Glycolysis.
change that pyruvate into something that can go into the citric acid cycle?
Are you talking the Krebs cycle?
The Krebs cycle and the citric acid cycle, they're the same thing. It's the cycle of many names.
Wait, are we talking about glycolysis or?
Glycolysis says that here.
We're trying to figure out where you're going.
Intermediate.
There we go, intermediate stuff.
Oh, okay.
And then we create a whole bunch of NAD plus or NADH and FADH2, and we send that to what to get more energy out of it?
The Krebs cycle.
The Krebs cycle makes it.
Oh, sorry. The
Repeat the question.
Okay, the Krebs cycle, the citric acid cycle makes a whole bunch of NADH and FADH2, and we send that to what to get more energy out of it?
Is it the cellular respiration?
The whole process from glycolysis to this last part is aerobic respiration.
Is it the ATP?
It does make a lot of ATP.
Listen to us go.
Like I said, you remember stuff better if you struggle to remember it. The electron transport chain.
Oh, okay.
Yeah. See where
I know
I'm
where you're
going here?
going now.
Okay, anyway, all of that's in the mitochondrium, and that's how we get a whole bunch of ATP out of these guys. Okay. Let me see if there's anything else. Oh, yep. We've got one more membranous structure in eukaryotic cells, just for the fun of it. Let's go ahead and do that one.
Do what?
There's one more membranous, membrane-bound structure. Tends to be close to the nucleus. Stuff that's made in the rough endoplasmic reticulum and the smooth endoplasmic reticulum comes over here. It's like pita bread or flattened pancakes.
Is that my Gucci bags?
It is your Gucci bags. What's its real name?
I don't know, but I just know them as my Gucci bags.
It is the Gucci bags. It
It starts with the G. I know that. Is it Golgi?
Golgi
Very
apparatus.
good. Yes, Golgi apparatus. Good job.
Woohoo!
Good job. And then I'm going to go ahead and stick in the cell membranes. I'm going to put some transporter proteins because that becomes important later. And do bacteria have transporter proteins?
Yep.
They do. They've got to have double sets. We've got to have some in the cell wall as well as in the cell membrane. Because, you know, we've got to get past two barriers. Okay. All right. Good job. Very, very good job. Okay. So, let's talk. We started talking about pathways. Okay. Do bacteria and human cells both have the enzymes for replicating DNA?
Yes.
Replicating what?
DNA. So when this guy gets ready to divide or this guy gets ready to divide, yeah, we've got to double our chromosomes. We've got to make more of our DNA. And do we have enzymes to make copies of the DNA that go over here to the ribosomes?
The bacteria doesn't, right? It takes all that stuff from the host? Or is that a virus?
Viruses. If you were talking about viruses, then yes.
Okay.
But with bacteria, yeah, they do. So do the humans. Yep, we both have RNA polymerase, which makes the RNA that goes into ribosomes, and also mRNAs that get read to make proteins, and then tRNAs. And so we share these enzymes. Depending upon the microbe, we may share some of these. Okay, if this is an aerobic bacterium, it may have the enzymes for the intermediate step, the citric acid cycle, the electron transport chain. Most bacteria and eukaryotic cells have the enzymes for glycolysis, so they have enzymes to make energy. Okay? So shared things, you know, that they both have, but they might be a little bit different. Okay, because it's been a long time since the shared ancestor between bacteria and humans. Okay, there's been a lot of separation through time. Bacteria have developed different mutations. They've developed different structures, and their enzymes are just a little bit different. Okay? Over here with humans, same thing. Okay, it's been a long time since we've exchanged DNA with bacteria. Okay. All right, we're feeling pretty comfortable with the structures of, say, a human cell versus a bacterial cell. Ready for me to ask you questions? Do some polls?
Sure, go for it.
Yeah. All right. Let's get a poll up. I think this is a 4-answer question for whatever reason. It seemed like a good idea at the time. Nope, it's 5. Let me switch this. Let's get 5 answers. There we go. Which of the following is a structure that bacteria have that eukaryotic cells do not have? And once again, it's okay if you don't remember for sure, because as you struggle to remember, your brain will say, huh, I've got to keep a hold of that answer when I get it. Okay, we're past 50%. Good job. We're at 71%. We're getting pretty close to 100. There we go. Good job. Okay, the favorite answer was flagella. Okay, and actually both eukaryotic cells and bacteria have flagella. They're just structured a little differently. So good job of remembering that they have different structures. Okay. But yeah, they both have flagella. So not that one. Our next favorite answer was 70S ribosomes. And yes, bacteria have 70S ribosomes. They're a little bit smaller than the eukaryotic ribosomes.
It's
So
the
good job.
nucleus, huh? Nucleus, right?
Well, if I switched it around and said, what structure do eukaryotic cells have that bacteria don't?
That's
then
how I
we'd
read
have-
it.
Yeah, then we'd have two right answers. Yeah. It's easy to get these kinds of questions backwards. Yeah. Yeah. So if I was asking what eukaryotic cells have, then it would be the nucleus and the 7 or the ADS. Okay, so good job. Okay, let's ask about, now I should have switched up the question so it was, what metabolic pathways do bacteria have that eukaryotic cells do not have? So we're thinking enzymes, metabolic pathways. Okay, we're coming up on 50%. There we go. Okay, we're
I'm
almost
not
to
sure
100.
why, but I don't see the poll.
Oh, you don't
I
see the
can-
poll.
I mean, I can see the question, but I can't.
Huh. Huh. Wonder what happened. Gotta love technology. Well, I'll tell you what, hopefully on the next one, you'll be able to see it. All right. So the favorite answer was electron transport chain enzymes. And depending upon what kind of bacteria, if they are aerobic bacteria or facultative anaerobes, then yeah, they're going to have electron transport chain enzymes. Okay. If they're anaerobes or aerotolerant anaerobes, well, instead of saying anaerobes, I should say obligate anaerobes. Then yeah, maybe they don't. So yeah, that's a good answer. It's not the best answer because you know, most eukaryotic cells have the electron transport chain enzymes, okay? Because we have mitochondria. There are very few eukaryotic cells that do not have mitochondria, and what they do have are mitochondria that have had mutations and have lost the electron transport chain enzymes. Okay, so not the best answer. So we have a three-way tie with enzymes that make peptidoglycan, Krebs cycle enzymes, and RNA polymerase. And to my mind, the best answer is the enzymes to make peptidoglycan, okay? Because eukaryotic cells never have peptidoglycan. We just don't have the enzymes to make it. We only see peptidoglycan in bacteria. Okay? So that would be the correct answer. Most bacteria have Krebs cycle enzymes. They all have to have some sort of RNA polymerase. Okay? So if you didn't pick enzymes to make peptidoglycan, don't feel bad. You'll remember it better. Okay, any questions about why the right answer was the right answer? Okay, let's go back to the whiteboard and let's talk about classes of antibacterial drugs and their targets. Okay, so when we talk about antibacterial drugs, generally we use the term antibiotic. Technically, antibiotic means anything that is against life, but in common usage, we mean antibacterial agents. Okay, and we have a large class of antibiotics that target peptidoglycan cell wall synthesis. Okay? So so they all target peptidoglycan formation some way. It just depends upon which enzyme. Okay. So there's one group that I want you to know. I'm going to toss in another one because you hear about it a lot. Okay, so penicillin. Okay, penicillin and all of the psyllins. Okay, they're part of what's called the beta-lactam superfamily. Okay. One of the first antibiotics to be developed and to be produced in a commercially available amounts, okay, originally isolated from mold in the penicillium family, which is why we call it penicillin. So all of the psyllins, amoxicillin, ampicillin, methicillin, oxacillin, okay, they're all in the beta-lactam superfamily, okay, and they all inhibit peptidoglycan cell wall synthesis. What's going to happen when you stop a cell from being able to make more cell wall?
Won't they lyse?
They will lyse. Yep. So one of the things to consider, though, is that it's mainly effective against bacteria that are rapidly dividing, which, you know, most of the time when you're talking a pathogen in the human body, they are dividing as fast as they possibly can before they get out. Okay. But there are some bacteria that are very slow growing and the beta-lactams are less effective against them. Okay. There's also another cell wall synthesis inhibitor, vancomycin. Okay, you notice it's not a psyllin, it's a myosin. Okay. Also originally, well, it was isolated from bacteria, but under the microscope, they look an awful lot like fungi. Okay. But this one also inhibits cell wall. Okay. You don't have to remember that one, but in case you've heard of vancomycin. Okay. So penicillin and the beta-lactams, generally we have very low side effects. Okay. Unintended reactions to a drug. Okay. And it's because we don't have cell wall. Okay. So these drugs that target the enzymes that make peptidoglycan, they're not going to bother us because we don't have those enzymes. Yeah, cool, right? But there are some folks that should not take penicillin. Okay, and they usually go wear a bracelet, a med alert bracelet. Why?
They're allergic.
They're allergic. Okay. So you can have the side effect of having an allergy to penicillin. Okay. Now this is my hypothesis as to why we would develop an allergy. Your body is going to see this and go, oh no, we've been infected with a mold. So you have an immune response to it because your body thinks you've been invaded by a mold. Now, there are folks that are not allergic, okay? And so, you know, not everybody develops an allergy, but we do keep track of allergic responses so that if you do have an allergy to it, we don't give it to you again. Because as we learned in the last couple of chapters, once you've been exposed to it, you have an immune response, you're going to have a stronger immune response the next time you're exposed to it. So if I have an immune response, an allergy to penicillin, it's going to be worse the next time I get it. Yeah, so we want to make sure that we don't give it to you. Okay? There's another unintended side effect, okay, from the penicillin family, okay? And relatively recently, we found out about this, and that's that antibiotics don't know the difference between the pathogen we're trying to kill and the friendly bacteria in your gut, okay? So it can lead to dysbiosis. Okay, the term from clear at the beginning of the semester, but what it means is that we get our gut community out of balance, okay, because we've got all sorts of bacteria dying left and right, and the yeast that are hanging out in small numbers says, I don't care, I have a cell wall, but it's not made out of peptidoglycan. This stuff doesn't bother me at all. And so they grow to higher numbers, and so you can get yeast infections. Okay. Sometimes you see diarrhea when you take antibiotics. Okay. All right. So, but generally speaking, with the beta lactams, with the class of antibiotics that target cell wall synthesis, specifically peptidoglycan cell wall synthesis, we have very low side effects. Okay. Any questions? Any questions about these guys? Okay, we have another group, okay, that targets 70S ribosomes, okay, and we call them protein synthesis inhibitors. And because they bind to 70S ribosomes and stop them from functioning, and when you stop the ribosomes, you stop protein synthesis, okay? And the example for this group that I want you to know are the tetracyclines. Okay, generally we call it just tetracycline. That's the most common member of the family. Okay. Also relatively low side effects. Okay. Because over here, we've got 80S ribosomes and they're just the wrong shape for tetracycline to bind to, okay? So it doesn't bother our ribosomes out here in the cytoplasm. Potentially, they could bother the ribosomes in our mitochondria, but generally speaking, tetracyclines will come in through the transporters, okay? But there aren't any transporters for tetracycline to get into mitochondria, so they just kind of bounce off. Okay? There are other protein synthesis inhibitors that could potentially get into the mitochondria and start killing them. Okay, so
Would that be erythromycin?
yes, you called it. Yep. Generally speaking, we only give that to you in the hospital so that we can track and watch out for what is the side effect that indicates that you have cells dying. You can get temporary deafness because for whatever reason, nerve cells are more sensitive. Yeah. But we do have side effects, unintentional consequences, okay, to tetracycline. If you take it, if you're pregnant, or if you are a child and you've still got teeth forming, okay, it can cause problems with teeth and bone formation. Okay? With young kids, it's just discolored teeth. We don't see that as much anymore, but it used to be, you know, if you ever see anybody that's got kind of gray teeth, and they're older, you know, 50s, 60s. It's probably because they got tetracycline when they were a kid. I have a friend that contracted whooping cough, and so they needed, and which is caused by a bacterium, and so they gave her tetracycline, and it discolored her adult teeth. But if a woman is pregnant, it can cause brittle bone formation. So we generally do not give tetracyclines to pregnant women because it can harm the fetus. Okay. And then you can also have allergies, and you can also have dysbiosis.
Don't you have photosensitivity too?
Oh, that's right. Yes, I forgot about the photosensitivity. And personally, I have no idea why that shows up. I don't know. But yeah, you're right. They tell you when you're on tetracycline to stay out of the sun because you can develop a rash and different things. Yeah, which is, sometimes with any drug, you just get these weird reactions. But relatively few side effects. Now, next time we meet, when we talk about antifungals and antiprotozoal drugs, yeah, we have more side effects because they're more closely related to us. It's harder to find unique targets. Okay. Let's see. There are some that target the replication of the chromosome. Okay, I'm not going to have you learn any of those, but they have relatively few side effects because the enzymes that replicate DNA in bacteria are just different enough that generally they'll leave ours alone. Okay, so like ciprofloxacin is one that's commonly prescribed, but we don't have as many antibiotics in that category. Okay, but another one that I want you to know, okay, is let's talk about B vitamins. Okay, so bacteria are able to make the B vitamin folic acid. And I forget which B vitamin is. It's B something. But they take substrates and they have a couple of different enzymes that convert that into folic acid. Then they take folic acid and they make nucleotides out of it. And then they proceed to make DNA and RNA out of it. Okay. So folic acid is very important to synthesis of DNA and RNA. And if you don't have DNA and RNA, you can't make protein. So indirectly, it inhibits protein synthesis. Okay? But we call drugs that inhibit this process folic acid inhibitors. Okay, now we need folic acid too, okay? But we humans do not have these enzymes, okay? We have to eat folic acid. And so we eat it, it's transported into our cells, it's taken to the nucleus where it's converted into nucleotides. Actually, I think that happens in the smooth endoplasmic reticulum. We'll draw that there, and then it goes to the nucleus. And then our cells can divide. Okay, so folic acid is very important. Well, these inhibitors, okay, the sulfa drugs, sulfonamide, okay, inhibit these enzymes. We don't have those enzymes. Okay, so it doesn't bother us. Doesn't bother us. Well, I don't know about you, but I was wondering, hey, well, if we inhibit this process, can't the pathogen get folic acid from us? Because we're eating folic acid either in our food or in supplements. Okay, they don't have transporter proteins. Okay, so they cannot take folic acid up from the environment. Interesting, huh? So if we shut down this pathway, they die. Okay. So as far as specific antibiotics, I want you to know. I want you to know penicillins, penicillin and all the other cillins, okay, part of the beta-lactam superfamily. They're cell wall synthesis inhibitors. They inhibit peptidoglycan synthesis, okay. I want you to know that tetracyclines, okay, that they bind to 70S ribosomes, okay, and inhibit protein synthesis, okay. And And then I want you to know the sulfa drugs, and that they inhibit the enzymes that make folic acid. Okay, okay, questions? Oh, and by the way, we have kind of the same side effects here, unintentional consequences. Some folks are allergic to sulfa drugs. So they generally have a med alert bracelet so that we make sure we don't give that to them. And then also you can have dysbiosis from that. Okay, you ready to have me ask you questions? There we go. I don't know why I animated these. It seems like a good idea at the time. Now let me switch this to a 4-answer poll. Okay, so penicillin targets which of the following? Oh, let me relaunch this. I think I clicked a few too many things. There we go. Now you can see the poll. Hopefully you can see the poll. Okay, we're 71%. Working our way to 100%. We're almost there. Okay, I think we stalled a little bit here. Let's go ahead and oh, there we go, 100%. Yay! Far and away the favorite answer was cell wall synthesis. Good job. That is the correct answer. True, if the cell pops, then it's not going to be making any protein, but that's kind of an indirect consequence. Okay. So good job. Okay, so my next question, what do tetracyclines target? Okay, we're past 50%, coming up on 100%. Okay, tell you what, we'll go ahead and end the poll. We're almost to 100%, not quite. Okay, favorite answer was protein synthesis. Very good. That is the correct answer. Tetracyclines bind to 70S ribosomes. And if you bind to 70S ribosomes and stop them from doing their jobs, then the cell can't make any proteins. Okay. The next favorite answer was B vitamin synthesis. That's an indirect consequence. Okay. If you're not making proteins, you're not making the enzymes that make folic acid, but that's kind of indirect. Okay. It directly targets protein synthesis. Okay. Same thing for nucleic acid synthesis. Okay. If you're not making enzymes to replicate your nucleic acids, then that's an indirect consequence. But we would go with protein synthesis as being the correct answer for this one. Okay, one more. Okay, sulfa drugs. What do they target? Okay, coming up on 100%. Okay, we'll go ahead and end the poll. Okay, we got a three-way tie between B vitamin synthesis, cell wall synthesis, and nucleic acid synthesis. Okay. Sulfur drugs directly target B vitamin synthesis, specifically folic acid. Okay. So this would be the correct answer. That's what it directly targets. But it indirectly targets nucleic acid synthesis, because if you don't have folic acid, you can't make nucleotides. And if you can't make nucleotides, you can't make DNA and RNA. But that's kind of further down the line. Okay. Same thing with cell wall synthesis. It's further down the line. Okay. If you can't replicate your DNA, you can't make RNA. Can't make RNA, you can't make the enzymes to make your cell walls. Okay. So I'm going to go with B vitamin synthesis is the best answer of the three. Okay. But good job. You did a good job on working to remember what you learned. Okay. That's a good part of studying. And on Monday, we got done kind of early. We're getting done kind of early today, too. So the question is, do you want to start on what I'm going to talk about on Thursday, antifungals? We can talk about antifungals, or we can be done early today. Take your pick.
I'd rather learn on because you might run out of time on Thursday.
Sounds
Yeah,
like
agreed.
a good plan. Yep, let's go ahead and do that then. Okay, let's start talking about antifungals. All right, so I'm going to clear my drawings here, and I'm going to have you walk me through a fungal cell, okay, and a human cell. In this case, because they're both eukaryotes, humans are going to stand in for animal cells. Okay. All right, so as eukaryotes, what is the structure that all eukaryotic cells have?
Cell wall?
One of them has a cell wall, one of them doesn't.
Fungal has a cell wall.
Yes, it does have a cell wall. What is that cell wall made out of?
Isn't it also made out of peptidoglycan?
It is not.
Oh.
Fungi are not bacteria, but that was
Isn't it made
that
out
was a
of-
good thing.
Isn't it made out of
Lipid?
curtain?
Uh-huh.
Kite.
Curtain?
That's
I
what it
think
is.
it's chitin. I'm just never quite sure how to, is this a ch like as in church? Or yeah, this is one of those funky Greek words.
I get
that
mixed
if I
up
spoke-
chitin and curtain all the time. 'Cause
I know, because you can pronounce them both ways. So, yeah, but I think it's chitin. Folks that have actually studied Greek, I think, have told me that one. Yep. So, fungal cells have a cell wall made out of chitin, and this is why antibiotics that target bacterial cell walls leave the fungi alone. Okay. Mold, yeast, doesn't bother them a bit. Okay. Now, what's underneath this cell wall? What do all cells have? You don't have it, you're not a cell.
lipid bilayer.
Lipid bilayer, yes. And do animal cells have one?
Yes.
Very good. Yes. They both have cell membranes. Okay, so what's a structure in here that if I were looking under a microscope, a light microscope, I'm going to look in there and I'm going to go, oh, yep, that's a eukaryotic cell. What's the karyote in eukaryote?
Nucleus?
Nucleus. Do both fungal cells and human cells have a nucleus?
Sure do.
They do. Yes, that's what makes a eukaryotic cell a eukaryotic cell. Okay, do they both have mitochondria?
No.
You would think not,
Oh.
but actually they do. Fungi have mitochondria. Yeah, they've got them. Do they both have endoplasmic reticulum?
No.
You would think not, but actually they do. I'm just going to put one set here on both of them. So yeah, they both have an ER. We're not getting very many unique structures, are we? So far, we just have a cell wall. Do they both have a Golgi?
Sure do.
They do.
Got my Gucci bags.
Yep, we got my Gucci bags. Yeah, they both have Golgi. Yeah. So the question arises, how in the world are we going to shut down, what drugs, what targets could drugs go after with fungi? Okay. Part of it is it's a good thing that generally speaking, we don't have a serious fungal infections. Okay. Most of the time, if people get a fungal infection, it's annoying but non-lethal, what we call, they're called dermaphytes. Anyway, they're skin infections. Athlete's foot, ringworm, the hair one on your scalp, yeast infections of the vagina or the mouth, uncomfortable, but, generally you're okay. Unless you're immunocompromised. That's one of the signs that you are immunocompromised is if you get systemic fungal infections, okay? Because generally speaking, they don't like us. We're a little too warm for them. That's why they kind of like, generally speaking, your skin is a little cooler. There are some yeast that have adapted to living in the warmer interior, but not very many. Okay? But there are times when we do need to fight fungal infections. Okay? And it's really hard. So we don't have very many antifungal drugs. Okay. Because it's really hard to target these guys because they're so similar to us. Our last common ancestor of all the eukaryotes wasn't that long ago, relatively speaking. Okay, our genes and their genes are more similar, okay, than they are for bacteria. But we do have some antifungals, okay? And most of them, okay, most of them target, believe it or not, the cell membrane. And they target a steroid, okay, specifically in the cell membrane, and it's called ergosterol. Okay, and it's a cousin to cholesterol. Okay, now cholesterol has gotten a bad rap. We don't like it floating around in our veins and putting our veins in our arteries. Okay, but actually it's necessary for the cell membrane to maintain its fluidity and its health. Okay, as human cells gush around and do all of their flexible stuff. Okay, so fungi have a relative of that called ergosterol, and we have some drugs that target ergosterol. But we can have cross reactivity with these drugs with cholesterol. Okay? So you have to be really careful when you give these kinds of drugs systemically, when you take it orally or by IV. Okay? And the one I want you to know is amphotericin B. Okay? And it's a drug that we do give by IV, only in the hospital. You can have severe side effects. okay, from
Don't you
it.
get toxic kidneys infections,
Yep.
right?
Yep, because when you get a lot of cell slicing, it tends to clog up your kidneys in your liver and your spleen as it's trying to clear stuff out. Yeah, so we have to be very, very, very careful. We do have some others that are applied topically. So the creams, you can buy creams for athlete's foot and stuff over the counter. Okay, because you just rub it on your skin, it doesn't get into your system, and you're killing mold, okay, fungi that are living in the dead layers of your skin. Okay, what they're after is they're eating the keratin, the protein, and it probably wouldn't be a problem, except for your immune system recognizes that there's something foreign there and you get an immune response, which is the flaking, the blistering, and the inflammation that you get from athlete's foot and other skin fungal infections. But we could take something that would be toxic to eat, rub it on your skin, wash your hands, you're good to go. Yeah. So yeah, don't eat antifungals, by the way. But that's the antifungal that I want you to know. Okay. Okay, questions. Questions about this one. Okay, let's talk about viruses. And that ought to take us to the end of class. Antivirals. We're getting more and more of them all the time, but it's been a hard go, okay? Because viruses, generally speaking, use our machinery. They use our enzymes, right? And they replicate within the cell. And they're all different. Yeah, that's also the frustrating part is they're all different. So an antiviral that's going to work against HIV is not going to be working against herpes virus. And antivirals that work against HIV and herpes virus are not going to be effective against, say, Ebola. Back in 2014, during the West Africa Ebola epidemic, out of desperation, because they were having, you know, if you got Ebola, you had a 50% chance of dying. Them's not good odds. Okay, it's better than the 90% death rate that we were seeing earlier before that epidemic, but still 50% is pretty bad. Okay. COVID ain't got nothing on Ebola. So people would give their Ebola patients anti-HIV medications in the hopes that it would help. Okay, and we saw a similar thing with COVID. People were giving their patients other antivirals in the hope that it would work. Okay, but generally speaking, we do not see antivirals for specific virus working well against another one. Okay, so antivirals tend to be what we call narrow spectrum. And that means that they only work against a specific type of pathogen. Okay, so broad spectrum drugs would be like penicillin. Okay, well, let's go with ampicillin, which is kind of the second generation psyllin. It works equally well against gram-positive and gram-negative, so we call that a broad spectrum antibiotic. Okay? Whereas there's some antibiotics that target a specific group of bacteria. So antivirals tend to be very, very narrow spectrum. Okay? And the antiviral that I want you to know, okay, is acyclovir. Okay, and you can generally tell the antivirals because they tend to have vir in the name. Okay, vir for viral. Okay, and the reason why I bring up acyclovir is because its mode of action is pretty darn cool. Okay, acyclovir is a nucleotide analog. So if I were to draw your average nucleotide, okay, I've got a sugar group. And I've got a base hanging out here. It's either an A, T, G, C, or U. Okay. If it's RNA, then it's got a U instead of a T. And I've got a carbon, and I've got a phosphate group sticking up here. And I've got an OH here that my next nucleotide is going to hook onto. So if I'm say replicating my DNA. And I drew that too far over. Let me move it over so it lines up a little better. Okay. So what's going to happen is my phosphate group here is going to hook on here through a dehydration reaction. Oops. I got rid of my O here. Okay. And then I can bring in another nucleotide and I can continue to hook them together. Okay. Okay, acyclovir is kind of like the top half of a nucleotide. Okay, it's got a base, it's got a phosphate group. Okay. And so when it hooks on, okay, say it's going to be hooking on to this one here. It'll hook on just fine, but I can't bring another nucleotide in. Okay. So what that means is as the virus is trying to make more of its genome, okay, or if it's trying to make mRNAs, well, no, this one is just a DNA nucleotide analog. Okay, it stops the chain. And if it can't make more of itself, then it can't spread to another cell. Okay, cool, right? So acyclovir is found, and it's an anti-herpetic. Okay, so it is against herpes viruses. Okay, so you find it in cold sore medication. Okay, once again, topical. Okay. We give it to patients who have HIV, which sounds a little funny because, you know, HIV is not a herpes virus, but it will prevent reactivation of latent herpes infections. Okay, so it'll prevent a systemic chickenpox reactivation, bad shingles, in other words. Okay. It'll prevent folks who are into AIDS, instead of getting just a cold sore, they'll get kind of a whole body rash from herpes simplex. Okay. We can prevent that from happening by prescribing acyclovir orally. Okay. And, you know, there are other conditions if you are on an immunosuppressive because say you're getting a bone marrow transplant, or say you're getting a kidney transplant, oftentimes we will give patients acyclovir to prevent reactivation during that first time period when they're immunosuppressed. Okay. To prevent these herpes viruses that once you get them, you boys got them, we can't cure it, but we can prevent flare-ups. Okay. So
Shingles too.
and shingles, yeah,
Shingles.
if you get a bad case of shingles, we'll often prescribe the acyclovir. Yeah. Yeah, so acyclovir is pretty good. So I want you to know acyclovir as our example of an antiviral. But what I want you to know about antivirals is they're narrow spectrum and they target viral enzymes. Unless they're like acyclovir and they are a nucleotide analog. Yeah. So that's mainly what we have as far as antivirals. Now, having said that, we've developed some cool new stuff that are anti-COVID. And one of the first ones that we came up with were monoclonal antibodies. And these, what they do is they target attachment. Okay, so what we do is we make antibodies against COVID spikes. Okay, this would be an example, you know, if I've got a bag full of monoclonal antibodies and I've got you in an IV drip, this is an example of passive immunity. Okay, so you're not exposed to the antigen, well, other than the fact that you're infected with COVID. But I'm giving you antibodies that we've made in the lab so that you can recover, because remember, on that first time that you're exposed to a pathogen, it can take up to a month before you've got enough antibodies to shut down the infection, which is a long time to be sick.
I
Okay,
did
you might not
those.
get better.
I did those.
Yeah, and did it work pretty good?
Well, I don't
Mhm.
know. Well, I had COVID, then I got over COVID, then I got COVID again. So that's why we went in
Yeah.
hardcore on that way.
And the problem is, and we've known this about coronaviruses for decades. For some reason, they can prevent development of memory cells against them. And we don't know how it does it or why some people it's going to do that to and other people can develop an immunity to it. So yeah, no, that doesn't mean that you're going to get it again. But that's why we gave you the passive immunity to help you fight it the second time through.
Yep, and it's not, you don't even know, you know, I didn't have any kind of side effects off of it.
Which is good. Because what we do is we get bacteria or yeast, you know, because yeast can make our stuff just as good as we can. We get them making these antibodies in the factory, and then we purify them. So you're basically getting human antibodies. And so it's kind of like getting antibodies from your mother while you're a fetus. Yeah, so we generally don't have allergic reactions to them. Yeah. Kind of cool, huh?
So antivirals, they inhibit the DNA replication.
Acyclovir does.
Acyclovir does.
Yes. Most of the anti-HIV drugs or the anti-Ebola drugs that we're developing target viral enzymes, enzymes that the viruses have that we don't. And then you also have the monoclonal antibodies, which prevent them from infecting new cells. Yeah, so like I said, antivirals are weird. You've got to do something different for each different type of virus.
Is valcyclovir the same thing then?
Yeah, valcyclovir is it's either a second or a third generation from
Okay.
acyclovir. Yeah.
So any
So
of
it's
the
in
virs,
the cyclovir.
basically?
Yeah.
Okay.
Yeah. So it's in the same family as acyclovir.
And they can be either oral or topical.
Yep. Yep. We have relatively few side effects from this one. Yeah. Hey, good questions. Other questions? Tell you what, let's go back to the PowerPoint and let's talk about what to do for next time. So as you can probably tell, for this chapter, it's good to review chapters 3, 4, 5, and 6, and chapter 8. So in other words, the chapters on cell structure and on viral replication and metabolism. Okay. I do want you to take the course learning objective post-assessment. For this assessment, what it is I'm going to compare this at the end of the semester to the pre-assessment at the beginning of the semester. Let's see how well you all did. I just look at it as an average, and if there's a certain course learning objective that I'm saying, yeah, we're not doing so good, I know I need to work on that more for the next semester. So please take this. If you get the questions right, you get 100% of the points. If you get it wrong, you get 75% of the points. Okay, so I don't want it to be high stress. It's timed, so you don't have a whole lot of time to look stuff up, because I want to know what you know. Okay. But there is incentive to do your best on it. Okay. Also, course evaluation. If 80% of the class does it, then those who do a course evaluation, I will give 3 extra credit points. Okay. And then also, I just found out last part of last week that that the IT department would like at least 75% of the student body to take the cybersecurity training. So I'm offering 3 extra credit points for that. Yes, Mitchell.
So when you say 80% of the class, how many students are in this section of the class right now?
I don't remember. I'll have to look it up.
Is there at least like 80% in the classroom right now?
Not everybody is coming to class anymore, but I do have at least 80% that are still doing assignments and taking tests. And so the 80% is, yes, 80% of those who are still actively involved in the class.
Okay.
Yeah. So tell you what, I will figure that out tomorrow. And then I generally do a ticker. I say, hey, we need this many more students to get to 80%. I haven't done that this semester. I think we're all tired. Just haven't been writing the test. But I will let you know how many more people need to take the or do a course evaluation and submit a screenshot. Okay, other questions?
I had a question. I was kind of struggling to figure out where to turn in the course evaluation.
Oh, let me show you. Let me show you where that is. I have a good reason for where I put it. It probably would make more sense to have put it in week 15, but I tend to put extra credit at the end, because it's extra. So if in lectures and assignments, if you scroll down to the week 16, then you'll see the link for the course evaluation and also for the cybersecurity batch.
Oh, awesome. Thank you.
All right, quite welcome. Yeah, probably would make more sense to put it in week 15, but it made sense to me at the time. Hey, any other questions?
The course evaluation, is that open now?
It is open. It will be open until Monday at 8 a.m. And you can submit your screenshot after that, but you won't be able to do the course evaluation after that, after Monday at 8.